It is my distinct pleasure to introduce the next presenter. In today's program, Dr. Naomi Hix. Who earned a PhD from the University of Texas in philosophy and a second doctorate from the University of Warwick in the UK. Dr. Hicksworth teaches courses in microbial poetics, the sonnet as a scientific method, and bacterial metaphysics. Her research emphasizes three realms. First, the evolution of language using the models of poems and nucleic acids. Next, how poems and microbial communities are structured by analogous processes. And finally, what comes from the gut in terms of both poetry and pathogens. He has published a number of important works, including 3 poems from the phylum Acetobacteria, provide insight into the meaning of microorganisms in soils, published in Applied Environmental Microbial Poetics, and a complete genome sequence of an aerobic poet, living as an endosymbiont in a department of molecular biology, published in the Journal of Things Money Can't Buy. Most impressively, Hicksworth was a National Book Award finalist for the enchanting work Chromatic, which was a qualitative exploration of postmodern microbiology in creative communities shaped by culture and environment. I should also note that Hicksworth won both the T.S. Eliot Prize and the Louis Pasteur Medal for Poetic Microbiology. And he received a National Endowment for the Arts and Sciences, you didn't know there was one of those, National Endowment for the Arts and Sciences Fellowship in support of her work on poems and plantomycetes. At present, Hicksworth is deeply involved in a project on the philosophy of language and microbiology called Why Doesn't Anything Rhyme with Bacteria? Once again, if you would like to disentangle this entirely true but factually disorganized introduction, I encourage you to visit the University of Wyoming's websites for the departments of molecular biology and philosophy. There you will find the genuinely remarkable accomplishments of Dr. Harvey Hix and Naomi Ward, who really have been finalists for the National Book Award and have published in the world's best and leading scientific journals.
Thank you, Jeff. And let me repeat the thank you to all of you for being here this morning. I'm a poet, and so this is terrifying. I've never had an audience before. So if you didn't know anything about either of us and you saw this image, we've been talking about the relationship between what we know and what we see and how we see. What do you see in this image? What do you think this is?
Bacterium.
Bacterium. Cheater.
Light.
Right.
Okay. Any other options? What do you think the, oh, sorry.
Holes from an interior showing the outside.
Okay.
Oh. What do the colors mean? Cool, they're cool. Color, temperatures, okay. Any other bids?
I like the idea that it's a view of the outside. I mean, these are natural colors. You got like the sky, and then you have the green that could be a plant.
Oh.
Right.
Okay.
Right.
So one more question about this image. What do you think the scale is? So, and the nurse is not allowed to answer. How these round objects, which may or may not be bacteria, how big do you think they are? Microscopic. Okay. in a unit of measurement.
They are deep space nebulae, light years across.
Okay. We'd like to take this image and put it into its more usual context. And we're going to do that by a process of zooming out over several stages. So first of all, we're going to go here. And now you can see that the original image is down in the bottom right. It's one of several that make up a composite. Some things that you can see have been added are some arrows and the word merge. And the latter is because the multicolor image is the blue, the red, and a green signal all layered on top of each other. Okay? So we're getting a little bit more information out of this image. We know there are different things within one unit, and they are located in different places. So at this point, the audience is thinking, 8 years of graduate and postgraduate training, and this is all she can tell us about this image. There's a little bit more. So we're going to zoom out again. So now you see, in fact, that that whole composite is only one of 3 panels in a figure. You also see a figure legend with a very great deal of text, including very long words and impossible to understand abbreviations. But we can pull some things out of this. So now we do, in fact, confirm that these are cells. They are cells of a bacterium called Gemata obscuriglobus. We see, as we suspected, that we're detecting things. And the things that we are detecting are called translating ribosomes. You're going to hear more about those a little bit later. I'm going to zoom out for the final time, and we'll see that this is actually one of many figures in a scientific paper, which you might have guessed. This is a paper from my research group. It represents about 4 years of work done by the 4 people that you see in that author line. Most of it was done by the first person, my graduate student, Ekaterina. So this paper is not yet published. In fact, it was accepted for publication by a journal yesterday morning. So I can honestly say that it's in press. So you are actually the first people outside of my research group to see it as a paper, which I think is really cool. Okay, so we are here with the paper, and what we are going to do next is look at that phrase, translating ribosomes. Okay, and for this part, I'm feeling a little bit intimidated, so I'm going to turn over to our resident microbiology, molecular biology expert, and that would be Harvey.
Yes, so I will now demonstrate the nearly infinite depth of my knowledge of microbiology that I've acquired in the last few days. So what you're seeing is a comparison between 2 kinds of cells, and it turns out that there are 2 kinds of, well, many kinds. This is one way to divide creatures in the world between prokaryotes, those who trip over slides, prokaryotes and eukaryotes. And so the distinction being made here is that bacteria are prokaryotes and you and I are eukaryotes, as are plants. So plants and animals, all eukaryotes. The kinds of things that we can see with the naked eye around us, all eukaryotes. And the difference between them that's relevant to the research that Naomi has been engaged in is that they make proteins in a different way. So in a prokaryote, in a bacterium, the purple strand that you see here is the information that was contained in the DNA, the information that tells the body what it is, how to reproduce, and so on. That information is transcribed on this purple ribbon, and then it's translated by the ribosomes. Just checking to see if she remembered. So the analogy is something like taking a plan or a blueprint and photocopying it so that it can be distributed to builders to make the thing from the information. And so the builders, the ribosomes, then make the proteins or the polypeptides, as they're called here, which is a nostalgic thing for me because I dated a girl one time, polypeptide. Yeah, I know. Ta-da. She dumped me like everyone else did. So the proteins then are made here. The difference is that the transcription, the passing along of the information, and the translation, the making of stuff from the information, happen at the same place and at the same time. But in a eukaryote, in you and me, the transcription, the passing along of the information, occurs here in the nucleus of the cell. And so the purple ribbon gets made here, but then it gets transported. And I was delighted to learn that it is sometimes transported by a chaperone. Go figure. It's sometimes transported by a chaperone, but in any case it's transported outside of the nucleus, and then in a different place within the cell and at a different time, then stuff is built from it. So a fundamental difference between the way bacteria operate and the way that you and I operate, or the way that our cells operate.
I have so much yet to learn from Harvey on this subject. So one thing I'd like you to note is the cell structures of the example prokaryote and eukaryote that we have in there. So just keep in your mind, coupled processes, simple cell, uncoupled processes, complex cell. And what I'd like you to do is look at the cell that I'm going to show you next and tell me whether it should have coupled or uncoupled processes. Any votes for coupled? Pretty unanimous? Okay, so that is a very logical conclusion because we see here the DNA of the cell. It's very condensed, it's very tightly packed. We see ribosomes out here and over there, and you can see that they are physically separated from the nucleoid by these membranes that look like roads or railroad tracks. Okay. So when I first saw images of this organism, which is Gemida obscuri globus, it seemed to me almost impossible that it could have coupled transcription and translation, even though it is, for absolutely definitely sure, a bacterium. So this is the 4-year project. It took us 4 years because it's a very uncooperative bacterium. It doesn't want to give up its secrets, but we beat it into submission. And we looked at those processes. And this brings us back to the image, which you are all now in a position to evaluate in a different way if I tell you that blue is a marker for transcription and red is a marker for translation within the overall cell structure that's shown in green. So you were right. It's uncoupled. And so that was the finding that's described in our paper. Multiple lines of experimental evidence suggest that there is uncoupled transcription and translation in this organism. So that was very satisfying. We're going to remove the annotation now, and we're going to come back to the pure image for the purposes of the art part of our art science undertaking. We'll go back over here.
So we started, obviously, in very different places, microbiology and poetry, another logical pairing, right? I think Jeff must have done all of the logical pairings, and we were left. But we, of course, as we began to work together, we noticed many, many points of contact between our disciplines and our practices. Many, many things that were shared preoccupations between us that begin with, for instance, the terms that are used in the paper. So the very title of the paper talks about the two processes of transcription and translation. So bacteria are poets too. I felt very comfortable. But as we discussed further, we kept noting other commonalities, common themes that also related to the larger discussions that the whole group had at our evening meals and other meetings. Such themes as constraint, which became very important for us in discussing our possible project, communication, process, wonder. We were interested in a number of things that have already come up as points of discussion in this morning's session. So I noticed the conversation just moments ago about being trained observers, and that both of us are trained observers, but we're trained very differently. And so we observe very differently. Charlie was just mentioning about a change in understanding, and Ashley's response about how that changes perception. We've spoken this morning already about movement, about shared vocabulary, about quantification, analysis, repeatability. We've talked about pattern, we've talked about balance, we've talked about dimensionality. So all of these things have to do with bacteria, with the microbial world, the world that's happening all around us and within us, but that we can't see. Similar to what's happening underneath the ground. We can't see it, but it's at work and it's very important. And so we tried to put these different preoccupations together and to construct for ourselves a working hypothesis. So our working hypothesis, I was opposed to the hypothesis, but I was bullied into it. So our working hypothesis was that if form and content are interdependent, if, in other words, how you look influences what you see, and if a form, such as the form of a scientific paper or the form of a poem, both shapes and is shaped by what the paper or the poem is about. So if form and content are interdependent, then if you translate content from one form to another, discovery should result. If they go together and you change one, you must have changed the other one. So that was our working hypothesis, that new content should appear as the result of a change in form. So our methodology then became to attempt to translate each of these forms, the form of a scientific paper and the form of a poem, into one another. A poem into a scientific paper into a poem. And so we'd now like to offer one of those sample outputs. So one direction for the kind of translation that we were interested in is from a scientific paper to a poem. For constraints, we decided on a particular poetic form that's called a glossa, which has these rules. They'll seem silly, but poets are that way. The poem opens with a 4-line stanza that's quoted from someone else, typically a well-known poet. Then the poem itself is made of 4 10-line stanzas. The last line of each stanza is quoted from that 4-line quotation. So it's just plucked and it's put in at the end of each stanza. And the 6th, 9th, and 10th lines of each stanza rhyme. Again, it's silly, I know. What? the stereotype about heavy drinking poets. So also each line in the poem is 10 syllables long, give or take, poems are cheaters. So this is a form that I have used before. There's a whole sequence of them in my most recent book. Though I should point out one way in which this poem cheats on the form. It cheated in order to suit the purposes of this experiment. So this poem, though it begins with 4 lines quoted from another poet, then the last lines of the stanza are actually quoted from Naomi's paper. So a little bit of cheating. So this poem translates Naomi's paper on bacterial transcription and translation by using the metaphor of written transcription and translation, the kind performed by monks prior to the invention of the printing press, when any reproduction of a written text had to be done by hand. It helps to know that the poem is making the analogy between transcription of DNA and transcription of a written human language by archaeological imagery. So the images of digging down through layers of a ruined city. So I'll save any further commentary or discussion for after the poem itself. The poem is called Transcriptorium, and here are the 4 lines by another poet. These are from the poet James Merrill. Then sky alone is left, 100 blue fragments in revolution, with no clue to where a niche will open. Quite a task, putting together heaven, yet we do. And here is the poem itself. A crumbled town, layers of dust and bone, streets spoke to a monastery down through soil horizons, but vellum bears traces of a difference. Deep layers yield transcripts and translations in the same hand, with the same blues and greens, deep and cold. Shallower, 2 different pens, 2 pressures, 2 blues almost like, but tilt the page and they flash different, one pale, one bold, conjugated to a smaller gold. The older strata describe a lone scribe with few brothers, all pray and chant and walk through moss-cool cloisters, but only from his pen do the books replicate and transmute to the common tongue. Rock yields pigment offspring, goethite yellow extracted, clinker welded hematite, cinnabars vermilion, miniature echoes of the icons, domes, spires, through his hand perfected. The volumes are all interconnected. Darkened lecture hall, the professor speaks of a later, larger town. Trade brought wealth, masters and apprentices from different lands, many crafts named in many different ways. Each sought their own words, too, for God and fear, for all the rules and their encapsulating. The scribes adapted, one wholly occupied with transcripts, 5 more outside the walls, a new colony, an illuminating location of actively translating. New ways, new rules. The extramural 5 were sisters, sequestered beyond simple diffusion of the texts by lay brothers in their errands about the town. Next were urchins, scampering chaperones who shed the metallurged fragments as they scattered dendroid, street distracted, which is why from later digs emerged a trafficking device. From hillside, abbot's perch to abbess, the books convoyed, a hollow rod or so-called solenoid. So we'll pass around the poem.
If you could scatter and some to the back.
So you'll soon have copies of the poem. Are there questions, observations?
It's very well crafted. Could you actually pick a couple of those out so we know specifically some of the, are they the last?
Yes, they are the last of each stanza. So if you're looking after the 4-line quote, then in the first stanza, conjugated to a smaller gold. in the second, the line, the volumes are all interconnected, and then location of actively translating, and then the very last line of the poem, a hollow rod or so-called solenoid. They're all taken directly from the paper.
Well, I guess I have to ask the scientists if this hypothesis has been supported.
From my understanding of the process, it's hard to convey all of the information in the paper, but I think that the gist of it is there.
Other questions, observations, thoughts about the poem? Yes.
Would going through this process with him and with this poem, would that affect, or could it affect the way that you teach your students?
Yes, it will. I've actually asked Harvey if he would consider coming to my classroom and us essentially doing this Naomi and Harvey show for my students. And for them, mostly for the purposes of learning how to communicate, but also for the knowing and seeing aspect, because I think through the process there was maybe a change in knowing the content and direction of the paper through the process of writing the poem.
We might now reveal that this change in thinking about the paper through the process of writing the poem all occurred within Naomi's head. She wrote the poem. I didn't write the poem.
What was your process? She writes mine, and was Harvey saying, good?
Harvey saw the poem in various stages, but all that he ever said was, "It's fabulous." I swear. We definitely discussed the form, because obviously this is not a form of writing that I was used to, a constraint I was used to working within. Other things about the process, the identifying and extracting the text from the paper that could be part of the poem was completely addictive. I went in there and there and there and there, and I had this list of 35 or so possible candidates, and then they got narrowed down to ones that would fit the 10-syllable, and then they actually were built first, and then the rest of the stanza was almost built backwards, particularly because of the rhyming requirement.
Which you're not used to from scientific
No, which I'm not
papers.
used to from scientific papers either. And the whole thing was done in about 3 days. So that was also interesting. Part of the process was the time factor.
I'd like to invite our Yale colleagues up very quickly.
You guys did an amazing job, and I think so far it seems like you guys had the hardest job. You know, for a microbiologist to come here and a poet to come here, I think those immediate connections are harder to find. And I was just interested in how you drew inspiration from this place.
Maybe one way to describe it is what we did was not so much about Ucross, but came from Ucross, because this would not in a million years have happened without the experiment. I mean, we have been on campus for 5 years overlap, something like that, and we've never even met. So this wouldn't have happened without Ucross. You know, when the poem was coming to sort of almost done, is when the clinker and hematite and all the rock references came in. And it's because I walked into the kitchen one day and Ron was there, and geologist, microbiologist, what do we start talking about? Meter of poems and libretto. So anyway, we digressed because I was really curious about whether I could use a line from my paper that had gold nanoparticle in it. I really wanted to use it. So I was asking Ron how, if you ground up a rock to get a pigment, how small would those particles be? And so we did a nano micro kind of talk. And then we just got more generally into rocks that give pigments. And then when I learned about some of the things that hematite is used for, I went away and looked and found that, yes, actually hematite was used and it's the main chromophore of red pigment, of ochres, for example. So it was used in the manuscripts, in medieval manuscripts, and it was used in the construction of tools for the illuminating as well. So it's kind of just passing through the poem, but it came from here.
And it's certainly the same thing for me as a poet. That my desk is at a window that looks out on this place. It's extraordinary. It's amazing, and of course the collaboration, the conversation, information couldn't have happened without this place as the gathering place and the source of energy.
I find it also so fascinating how these projects come together, and then how you talk about them, and how everyone who's come up has such a different teacher persona, but so, I mean, you want to take all of these classes. So I would just say, thank you for inviting us into your classroom as you're learning all of this.
Okay, got to be honest here. When Harvey finished the poem, how many people believed that he was the author?
Yay, it worked.
Yes.
Is there a nomenclature? Is there a narrative style that a scientific article has to go through? Or will this affect you? Will you think of words differently as you assemble your next piece?
I don't think in a literal way, so rhyming every 6th, 9th, and 10th line. Well, it would be fun. I could do it, and probably no one would notice because they're not looking for it. But when we were still trying to disguise the authorship, and I answered the question about knowing and seeing, an actually really freaky thing happened when I was writing the 4th stanza. And I was stuck between 3rd and 4th, and I didn't know where to go with it. And I thought about various things and rejected them. And in the end, I came to the 4th stanza is about if the processes are uncoupled, how is the message getting to where it needs to be? And in the concluding sections of the paper, we outline these 3 possibilities: simple diffusion, chaperones, and a trafficking mechanism. So all of those appeared also in the 4th stanza. And then after I'd done it, I thought, oh, because I've been struggling with where to go next with this project. And I've been thinking about different options, and none of which were really exciting to me. And then thinking about why did I choose that made me think, okay, that's what I really want to know. That's the question. And that was the freaky part. It's poetry. It's gotten in there.
It does point out a common theme that, again, we've heard now in really all the presentations, and that is that each of us is engaged in we employ different means, we have different backgrounds, but all of us are trying to see whatever it is, and in whatever way, will allow us to see what we can't see. So this image, you can't see this. This is constructed through the aid of technology and expertise that she knows how to use. So she knows the bacteria had to be altered so that, how did it happen? Antibiotics get injected into the bacteria so and they've been dyed, so they'll attach to the nucleus so that it shows up as one color. And so on. So this is why we read the poem and not the scientific paper that resulted.
Since this is uncoupled, what are the ramifications when you try to relate that to the contagion of, I don't know if this bacterium is contagious? Does it cause illness?
Disease causing?
Yeah.
No. Unfortunately, for funding purposes, this organism does absolutely nothing of any relevance to mankind, as far as we know.
So is this the only one that's uncoupled, or have you found others
That's a really good question.
that are contagious?
As far as we know, this is the only bacterial species out of thousands that has this cell organization. So we would expect that it's also the only bacterial species out of thousands that does this. However, the bacteria that we study are heavily biased by us and our interests. So the culture collections that we have are vastly dominated by organisms that either kill us or help us.
Right.
Okay, so these ones that do neither, we don't have as many of them, so I don't know that we can say that it's not out there because we haven't been looking for it.
So, images like cloister, scribe, monastery, heaven are evocative, obviously, of both the spiritual and the ancient. And I'm wondering if you could comment on that relative to your fascination with microbiology.
Oh my, okay. So at one point when the idea of the poem first, or the making this connection came to me, I actually the next day accused Jeff of putting some mind-altering substances in the water, because what had happened was I had gone to take a nap, naps happen at Ucross, because we stay up really late, and I was thinking about the paper, how to make the paper a poem, and all of a sudden out of nowhere, the dissolution of the monasteries, that phrase came into my head. Too much watching of The Tudors, maybe, that HBO series, because Henry VIII this is what he did, and I started thinking about when they stormed the monasteries and they ransacked them and all the wealth went to the king, what were they taking? Well, they were taking precious metal objects, but they also took manuscripts because manuscripts were precious. And then I started thinking about the colors, and then the colors in the paper, and the transcription translation, and then
One of the reasons why I chose these people was because of a deep sense of humility that they had. They were willing to admit that they didn't know things. And sometimes that humility gets in the way. Because Naomi's paper is being published, but it's being published in one of the top 3 science journals in the world, the Proceedings of the National Academy of Sciences. She sort of glossed over that. So this is a big deal.
The Ucross-Pollination Experiment was sponsored by the Ucross Foundation, the Wyoming Humanities Council, Saturday University, the UW Biodiversity Institute, the UW Department of Philosophy, the UW Program in Creative Writing, and the UW Institute for Humanities Research.